Hello, everyone. My name is Maria Villalpando. I am a researcher at the Dominican Foundation for Marine Studies, or Fundemar for short. I will be presenting in place of the Executive Director, Rita Sayares, about how we successfully incorporated an assisted coral sexual reproduction program in the Dominican Republic. Fundemar is an NGO based in Villalpando in southeastern Dominican Republic. Villalpando is a coastal town with deep roots in artisanal fishing and has recently become a tourism hotspot, receiving over 1 million visitors per year. Our mission is to conserve local coastal marine ecosystems and resources, and we do this through two main programs, a marine mammal protection program and a marine biodiversity program. The latter focused mostly on coral reef conservation activities, which I will be talking about today. We all know that around the world, coral reefs are rapidly declining due to global and local pressures, and southeastern Dominican Republic is no exception. We have overfishing of reef herbivores, high levels of use due to tourism, and we have registered massive coral bleaching events in 2019 and 2020. Recently, we have observed potential signs of stony coral tissue loss disease as well. We now know that in tandem with lowering CO2 emissions, it's important to implement various coral restoration strategies to complement each other and give corals a better chance of surviving. Assisting the sexual reproduction of corals has become a pillar of coral restoration activities due to its potential benefits. It is not only a cost-effective and feasible way to produce large numbers of corals, but it can increase survival during early life stage bottlenecks and can therefore promote a higher genetic diversity in struggling coral populations, giving them the genetic tools needed to adapt to a rapidly changing environment. Starting in 2011, our coral program concentrating on monitoring reef health and growing Acropora cervicornis tissue in nurseries and transplanting that tissue back to the reef. In 2015 and 2016, we carried out pilot trials to integrate assisted coral sexual reproduction into the existing program. And these first trials were done with very basic rudimentary equipment, and they succeeded, and that motivated us to scale up these efforts. Fundemar was the first institution implementing these techniques in the Dominican Republic, and in 2019, we established the first larvae culturing facility in the country. Today, we are sharing with you all three components that were key for us to incorporate these techniques as a permanent program in our institution. This first key component is the existence of a robust coral restoration program. We worked for years creating self-sustaining mechanisms that allowed us to not only rely completely on winning grants, and when we actually won grants, we used that to improve and scale up the program. In Villalpando, we created a structure beneficial to both the local tourism industry and Fundemar through coral nursery adoption, by establishing successfully 8 Acropora cervicornis underwater nurseries, hosting massive transplanting events, such as the one you can see in this slide, the picture in the slide, and providing tangible program results. We established a good reputation amongst local authorities and created trust between partners. As a result, local authorities support our work with permits, and private organizations provide valuable in-kind support to our marine activities with boats, diving equipment, tanks, and personnel time. This robust structure allows us to invest time and resources into exploring novel techniques that could be integrated into our program in alliance with international organizations. The second key component was to create the infrastructure to rear the larvae. Seeker International, the Nature Conservancy, and Corallium Lab were essential in this process. With their support, we trained our staff during workshops and received the basic knowledge to replicate these techniques back home. Initially, we focused on collecting as much data as possible on the spawning time of our local corals. During this time, we also reflected on the benefits of having a land-based facility for larval rearing, especially for research projects and as a safeguard in case of storms or hurricanes. And also Seeker International would provide us with their innovative coral rearing in situ basins or cribs, allowing to easily cultivate large amounts of corals in the water in situ. And by combining land-based and in situ technologies, we would produce a large amount of settlers. Using the Corallium Lab in Mexico as a guide for designing a cost-effective land-based facility, we started to think about possibilities in our local context. Since our office space was already small and the facility should be close to the coast for water pumping, we thought that a mobile facility inside a container would be the best option for us. Challenges were designing the lab specifically for larval rearing and finding grants to fund construction, both of which we were able to do thanks to our alliances with international partners. Here is an overview of the wet lab design. The mobile lab was placed in front of the coast to easily pump seawater to the lab's reservoir, as you can see here with the arrow, where the water is filtered and sterilized before going into the aquariums. We have 4 aquarium systems. Here you see 3 in the picture, now we have 4. Each system has 3 tanks with their own circulation system. And the lab is capable of holding over 1,000 settlement substrates. And we try to integrate systems that automate maintenance work as much as possible, such as plumbing systems to refill and drain the tanks, water flow and filtering systems for the aquariums, chillers and coolers to control water temperature, and live updates of water temperature. Just briefly, some results that we had in 2019 and 2020 using the lab, the land-based facility, and the Secor cribs. We successfully produced sexual recruits from 5 species, from Diploria labyrinthiformis, Dendrogira cylindrus, Acropora palmata, Acropora cervicornis, and Corpophilia natans. In total, for both years, we produced over 4,500 settlement units with approximately 268,000 coral recruits that were sitting in reef areas, totaling around 1,880 meter squares of reef. The third and final key component is producing good science. So since this is a fairly recent field of study, we didn't want to just replicate techniques, but also contribute to the scientific knowledge on the subject. One example is that we grew settlers of the rare Dendrogira cylindrus in situ nurseries. You can see that in the picture on this slide, something that had not been reported before. We also adopted an experimental design framework and standardized monitoring protocols. For this, first, we clearly formulate hypotheses for the restoration experiment based on the comparison of variables, measuring experimental, reference, and control plots to effectively estimate restoration success. We then establish a priori the sampling efforts based on power analysis. Currently, we are interested in answering questions regarding the differential settlement and survival between culturing systems, substrate types, and coral species in our local context. Overall, our goal is to improve the short and long-term survival of coral settlers. We hope that these techniques can continue to expand and be replicated in other places in the Caribbean so we can collectively give corals a fighting chance. Thank you all for listening, and let me know if you have any questions. I leave you with this really cool video of our lab in Valladolid and the amazing team behind all this work, and also with the information from the executive director and our social media info in case you want to contact us. Thank you so much.